Hello everyone, so my name is Lisa Ileanausit. So today I will present to you about Ichthyoplankton that we get during our field trip at Pantai Pandan Lundu. So the introduction to Ichthyoplankton are eggs and larvae fish that found mainly in the upper 200 meters of the water column. And then the fish larvae mostly found in the shallow water estuaries area and the coral area due to high food supplies and to avoid the predators. Other than that, the fish larvae mostly found in the continental shelf is that because it was dragged by the wave of the ocean water. The process of collecting Ichthyoplankton data. So Pantai Pandan Lundut Sarawak has a variety fish that we can get and has a lot of place to stay nearby. Collecting sample during the sample collecting. The wave a bit calmer and the tidal height is around 1.8 meters. And we use the SIN net to collect the sample. Sample preservation, we use buffer formalin to preserve the sample by adding pure formalin to the seawater directly at the sampling site. Lab work, we we managed to identify two different fish, larvae species, and the total sample we have, which is five fish larvae. So this is the video from.
Takut jatuh, Mat.
So as we can see here, the water is a bit calmer. It is around our waist, and then we record the GPS when we start, and then when it reach the a minute and 30 seconds, we turn, and then we go back to the shores. Okay, we proceed. So for sample preservation, we make the buffer formalin by adding 100% pure formalin into the seawater at the sampling location. After that, we take the bottle sample and then to put the sample from the scene net, which contain buffer formalin in the bottle sample. After that, we proceed with the lab work. We use stereo microscope to analyze and categorize the fish. So in here we can see the five fish that we caught: 11.01, 13.54 from above to top, above top to bottom, and then 10.66 millimeters and 10.82 millimeters from right to left, and then the single fish here is 4.32 millimeters. So the family, which is Clupidae and Ambasidae, the total individual is four for Clupidae, and Ambasidae is one. So this is the first species, which is from the order of Clupiformes. Um, the family is Clupidae. The what we can see here, we can categorize um, by the dorsal fin and the anal fin, which they are not overlapped and the head is not compressed as the other family. And then the anal fin does not extend to the counterpeduncle, also which we can compare to the other family from the same order. This is one more fish larvae that we managed to get the picture. So from here we see that clearly it has the dorsal fin and anal fin, but anal fin are not too visible as the first picture. In here we can see the anal fin very bright in color, which is in here we cannot see it much, but we can tell it is not overlap. And then this is the second species, which is in the family Amasidae, and this fish was easier to be categorized between the five out of five individual fish larvae that we caught because of the empty gap on the head of the fish, and then the round eyes, and it has a small mouth which is um, not over through the fish eyes. And then I guess that's all about the fish species, the fish larvae species. For the challenges that we go through is that we took the sample twice, because for the first time we get the sample, we only caught a lot of the shrimp, uh, and we get none of the fish larvae. So we need to take another more another more sample, and we go use the CNET once more, and then take the second sample. Yeah. And then take the second sample. For that, I guess that is all from me. Thank you.